Hello, my name is Catherine Gannon, and I will be presenting on the M protein. The M protein was discovered by Rebecca Lansfield, who was an American biologist, in 1928. So what is M protein? The M protein is a virulence factor of Streptococcus. It was the first molecule on Streptococcus pyogenes to be completely sequenced, and is therefore considered the archetypal surface molecule for Streptococcus pyogenes and other gram-positive bacteria. It is a coding for group A streptococci and determines the type-specific immunity of the bacteria. There are over 200 serotypes of M protein, and someone can be infected with multiple strains of M protein during their lifetime. So what is Streptococcus pyogenes? Streptococcus pyogenes is also known as Group A Streptococcus. It is a human-specific pathogen. The most common streptococci are acute pharyngitis, strep throat, and impetigo. The pharyngeal infection may become acute rheumatic fever, which can cause cardiac damage in the mitral valve. There are 517,000 deaths worldwide each year due to Group A streptococcal diseases. When does rheumatic fever occur? Acute rheumatic fever occurs when Group A streptococcus is present, there is streptococcal antibody response from a recent infection, the organism persists, and the infection is in the upper respiratory tract. What does M protein do? The M protein allows the streptococcus to survive in infected tissues and avoid the human immune cells that try to kill it. M mutants, which are streptococci without the M protein, are unable to survive in phagocyte-containing blood. The M protein has anti-phagocytic functions that are critical to its survival in human tissues and fluids. The M protein helps Streptococcus pyogenes resist being ingested and killed by phagocytic cells in the blood of a non-immune host. M protein immunity. When a host is immune, there are type-specific antibodies against the M protein that can neutralize its anti-phagocytic effect. Phagocytosis then occurs, and any streptococci that have the same serotype M protein are killed. How does M protein work? Streptococcus pyogenes is able to persist in infected tissues because of the M protein. The M protein gives Streptococcus pyogenes the ability to resist phagocytosis by polymorphonuclear leukocytes in the absence of type-specific antibodies. If someone has a resistance to a Group A streptococcal infection, there is direct relation to the presence of type-specific antibodies to the M molecule. Let's learn more about the structure of the M protein. The M protein is a fibrillar protein that occurs as a dimer on the bacterial cell wall surface. The M protein is covalently attached to the rigid peptidoglycan layer. It is composed of repeating covalent structures which have protective antigenic determinants. The M6 protein has 4 sequence repeat domains, with different sizes and sequences. The region of the M molecule that creates type specificity is limited to approximately 50 amino acids on the N terminus. The Cunningham experiment. Cunningham's experiment aimed to free the type-specific M antigen of non-type-specific immunotoxic properties. Purified M protein preparations were subject to enzymatic digestion in dilute solutions of pepsin at a pH of 5.0. This greatly reduced the capacity of M protein to react with the non-type specific M antigen complement fixing antibody that human serum normally contains. The Perez-Casal experiment. J. Perez-Casal deleted M protein from a chromosome and then reintroduced M protein to that chromosome. This was called the M knockout mutant, and it did not survive in human blood because it did not contain the phagocytes, even though it had all the secretory molecules of the wild-type organism. Let's finish up this presentation with a little bit of true or false. First question. The M protein is a virulence factor of Streptococcus pyogenes, which is a pathogen that affects all mammals. If you said false, you are correct. The M protein is a virulence factor for Streptococcus pyogenes, which is a pathogen that only affects humans. True or false? The M protein promotes phagocytosis. This is false. The M protein has anti-phagocytic properties. True or false? The M protein is a fibrillar protein that occurs as a dimer on the bacterial cell wall surface. This one is true. This is the structure of the M protein. Thanks so much for listening to my presentation, and here are my sources.